The first step in mitosis is prophase. Prophase is the longest phase of mitosis. The chromosome will condense. There are three components to the mitotic spindle fiber, which plays a key role in the stage of prophase. The first are asters. Asters are short microtubule fibers that help attach to the opposite ends of the cells. There'll be these little things right here. The second is the kinetochore microtubules. These will help connect the cells. The kinetochore microtubules will be on the ends of the cell of the mitotic spindle because they are helping to, as I said, attach kinetochores to the cell. The non-kinetochore molecules are going to be these in the middle, which is the third component of the mitotic spindle fiber, and the non-kinetochore microtubules will elongate the cell as it prepares to divide. The next step, which is like a half step, is prometaphase, which is like the second step to prophase. In this stage, the nuclear envelope breaks down in order to get the chromosomes. The kinetochore microtubules of the spindle fibers attach to the kinetochores on both sides, and the non-kinetochore microtubules will attach to each other on both sides, so from this side and from this side. In this phase, the cell is getting ready for the next phase, which is metaphase. In metaphase, the chromosomes are attached to the kinetochore microtubules on both sides. So everywhere right here, they're attached to each other. Right here. And the chromosomes will go to the middle of the cell. The sister chromatids are being prepared to split into chromosomes for each of their daughter cells. In anaphase, the kinetochore microtubules pull the sister chromatids apart, each going to their own side. These will go over here and these will go over here. The kinetochores are gonna shorten in length as they're pulling them down. And so now we know that instead of the sister chromatids, they are now chromosomes. As they're pulling to each side, they are getting prepared to be split into their daughter cells. In telophase, the cell is basically undoing all that happened in prophase. All of the destruction that happened is basically getting repaired. We're gonna have DNA that's gonna recondense. It's gonna go back down to a small version. We're gonna have the nuclear envelopes form for each daughter cell, and nucleoli will appear. It is getting ready for the split. The last step of mitosis is cytokinesis, where it is just dividing the cytoplasm, which will then give you the two daughter cells. So in animal cells, there's a cleavage furrow that helps to separate the daughter cells, and it is a ring of actin that helps pinch the cell into two. It is different for plant cells. In plant cells, since there is a cell wall, the plant can't use the cleavage furrow, so they will use a cell plate that forms to separate the two cells into two daughter cells. Through the mitosis lab, I learned the right order of the cell cycle, how prophase, metaphase, like how they go in order. And I also learned about how the spindle attachments work to elongate and then shorten up again. I learned more about the kinetochore microtubules, which before I did not know about. In simple diffusion, a solute will go from high solute concentration to low solute concentration down the concentration gradient. This meaning that there is no need for any energy put into this task in order for it to be done. The solute has the tendency to go from high concentration to low concentration. These are some examples of how a solute will go from high concentration of solute to where there is none until it reaches equilibrium, where therefore it should be equal solute concentration on each side. With osmosis, I learned that it is diffusion of free water across a semi-permeable membrane, meaning that the solute does not move. However, the water does. So in a situation like this, the solute is not selectively permeable, however the water is. So the water will move where there's a high concentration of solute because it wants to like water it down. With osmosis, there comes 3 different ways that water can affect a cell. This is called tonicity. This is where we get those terms hypotonic solution, isotonic solution, and hypertonic solution. So a hypotonic solution is when there is higher solute concentration inside the cell than outside of the cell. Therefore, the water, all of the water will have the tendency to go in the cell, causing it to burst. Isotonic solution would be equilibrium, so the solute concentration is the same on each side. Hypertonic solution would mean that all the water in the cell would want to go out of the cell due to the fact that there's more higher solute concentration outside of the cell, causing it to shrink. Through the Symbio lab, I learned the difference between hypotonic and hypertonic solutions. For this, I never really could differentiate between the two. However, now I see that hypertonic solution is shrinking because, as I said before, there's more solute concentration outside of the cell, therefore water will leave, and hypotonic is the opposite of that. I learned the true meaning of osmosis, and what I mean by this is that I never really understood what osmosis was. I knew it had to do with water, but I didn't know what it was. And now I think the lab really helped me to see how water moves, and it's really not based on the solute. I mean, it is based on the solute, but the water moves, not the solute. It's all about how the water movement is. For the tissues, for the first tissue, I picked that it was a hyaline cartilage connective tissue. I picked this because it looks jelly. I think this is due to the collagen. And I can also see like the little dots, which are the chondrocytes. For the second one, I picked pseudostratified ciliated columnar epithelial tissue. I picked this because it looks like many layers. The nuclei aren't lined up, as well as the goblet cells are visible, and the cilia, you could see it. And that will be all for this video. Thank you.